Last lecture we talked about respiratory pigments that are found in animals, and the first, foremost, most common is hemoglobins, and the second are hemocyanins. And then we also talked about hemerythrins and chlorocurins as the last, least common respiratory pigments that are found in animals. Now keep in mind respiratory pigments and their presence in bodily fluids in animals is going to allow for an increase in oxygen carrying capacity of the fluids of that animal. So today we are specifically going to talk about the oxygen binding characteristics of respiratory pigments, and which ultimately will lead to certain levels of oxygen in bodily fluids. And it all is in relationship to the partial pressure of those fluids and how saturated those pigments might become. Now, just to start, respiratory pigments, besides increasing the carrying capacity for oxygen in blood, tissues, or other types of fluids, they also have been found to have different types of roles as well. They are important in sometimes storage of oxygen, which is what myoglobin is thought to be involved in skeletal muscle. They also have been found to buffer the blood pH. They can actually pick up hydrogen ions when the pH drops, and they can release hydrogen ions when the pH increases, and this is based on their deoxygenated state and also their oxygenated state. They also are important in transporting CO2, which helps us ultimately remove CO2 waste from tissues out of the body through the lungs. They've also been found to be important in facilitating oxygen diffusion through the cytoplasm. This is in a role of myoglobin, and other roles as well, such as enzymatic activity, and also some different types of roles depending on different organisms, such as animals that live in thermovents. The respiratory pigments have evolved to bind to something other than oxygen and help carry something like sulfides for the symbiotic bacteria that live in those animals. So ultimately, these respiratory pigments not only have one function, but have evolved to have many types of functions for different types of animals, and it just depends on the type of respiratory pigment as well as the animal in which that respiratory pigment occurs in. So when we think about oxygen binding to these respiratory pigments, let's talk a little bit about the binding characteristics of these oxygen to respiratory pigments, and ultimately what is the binding or carrying capacity at different partial pressures of oxygen in the blood. So when we look at the graph in front of you, what you'll notice is that the blue line is essentially telling you the percentage of heme groups that are oxygenated, so that are basically bound to oxygen. And you'll notice that the binding of oxygen to these heme groups in the blood essentially increases with increasing partial pressure. So as partial pressure of oxygen in the blood goes up, then obviously the hemoglobin molecules or respiratory pigments like hemoglobin would be essentially picking up or binding to more and more oxygen, till eventually there is what's called a saturation point when we get to that 100%. Now, just to remind you, in bodily fluids containing respiratory pigments, there is a large population of oxygen binding sites due to the presence of those respiratory pigments. And for example, the human blood has 5.4 times 10 to the 20 heme groups per 100 milliliters. So the presence of these respiratory pigments greatly increase the oxygen carrying capacity of the blood. So just from this slide alone, we can see that as partial pressure increases, more hemoglobin molecules are bound to oxygen until basically the blood becomes saturated with oxygen, so that all those binding sites are saturated. An alternate view, looking at the amount of oxygen in blood, we can look at the quantity of oxygen per 100 milliliters of blood, so the percent volume. And this is different than looking at just the saturation of hemoglobin in reference to oxygen carrying capacity, but actually look at the amount of oxygen per 100 milliliters. And so what you can see when saturation is reached, you can see that the saturation point is at 20 milliliters of oxygen per 100 milliliters. So the typical kind of carrying capacity or saturation point of oxygen in human blood is essentially at 20 milliliters of oxygen per 100 milliliters of blood. And you can see to reach that saturation point, we're looking at a partial pressure of about 100 millimeters per Hg, or a partial pressure in kPa's, which is a different way of looking at partial pressure as well. So something above 12. So we're looking at more 13, 14 kPa's, which is close to that 100 millimeters Hg. Now let's look at something in reference to the body. So the lung, when we look at the lung and the partial pressure of air in the lung, in the alveoles, it's at 100 millimeters Hg. And the arriving blood to the lung is going to be obviously at a lower partial pressure. It's going to be partly deoxygenated and essentially needs to pick up oxygen to deliver it to the rest of the body. When that blood leaves the lung, so as oxygen leaves the lung and moves into the blood, when that blood leaves the lung, is the partial pressure of the blood leaving the lung is at 95 millimeters Hg. So it's almost to that 100 millimeter Hg partial pressure, which means we're going to be very close to that saturation point, meaning that 20 milliliters of oxygen per 100 milliliters of blood in that blood when it leaves the lung. So it's going to be pretty close to saturation point, meaning all those heme groups are going to be saturated or pretty close to saturation. So now let's look at the tissues in general. When we think about tissues, extremities, ultimately the oxygen partial pressure is going to be lower at the tissues than the actual blood, the blood moving to those tissues. So the blood from our arteries. And essentially, this is going to be important because that lower partial pressure in the tissues is ultimately going to help drive movement of oxygen into those tissues. It's going to move from the high partial pressure of the blood into the tissues and into specifically the mitochondria, so ultimately aerobic respiration can occur. Because oxygen is readily used in aerobic respiration, it's eliminated almost instantaneously, so the partial pressure in the tissues is always going to stay lower, which is going to help be a huge driving force for movement of oxygen into cells. So essentially aerobic respiration can continue to occur. And to allow for that movement, the oxygen will unload, will basically be released from hemoglobin and will diffuse into those cells and ultimately be utilized by the mitochondria. So how can we measure how much oxygen is being delivered to the systemic tissue, or how much oxygen is being used, right, by the systemic tissue in general? And that ultimately is going to be how much is delivered to that tissue. And one of the easiest ways is to look at what the saturation of the venous blood is, meaning the blood returning to the heart that's going to be pumped to the lungs so that it can ultimately pick up oxygen. That blood that's carried by the veins, what's its saturation? So what's the level of the carrying capacity of the oxygen of the blood? Or in easier terms, how many milliliters of oxygen per 100 milliliters of blood is present in that return blood? And one way to do this is ultimately to look at the return blood in the veins, and ultimately you can get a mixture of the venous blood from all parts of the body by essentially drawing blood from the great veins that lead back to the heart. And that's going to have a mixture of blood from all different areas of the body that ultimately has certain amounts of oxygen that was released into those tissues, and the return blood basically has the oxygen, vacant that basically was used by those tissues. So there's going to be less oxygen in the blood returning to those great veins. So ultimately at rest, when we look at what we find in this mixed venous blood, this blood found in these great veins, the partial pressure of the blood is down to 40 millimeters Hg, or in other terms, it's down to 15 milliliters of oxygen per 100 milliliter of blood, of venous blood. And that would give you a 500 milliliters of oxygen that was released per 100 milliliters of blood to those tissues. And this is on average what we'd see at rest as far as usage or release of oxygen, 5 milliliters of oxygen per 100 milliliters of blood being released to the tissues. And the return blood is just going to be missing 25% of its blood, its oxygen carrying amount. Now, if we look at the same type of figure, when we look at our amounts, so we are saturated when we're at 20 milliliters of oxygen per 100 milliliters of blood. And when that blood returns to the veins, to the heart, in somebody that's at rest, this is the amount of oxygen that's released to those tissues and is no longer being carried by the blood. So 5 milliliters per 100 milliliters of blood. Now, what happens to this remaining amount of oxygen in the blood? Well, this is what's called the venous reserve. And ultimately, the venous reserve can be utilized in extraneous situations such as exercise. So it's additional oxygen that can be released under certain conditions such as exercise. But what you'll notice if you look at the release of oxygen based on this curve, and you'll notice the first 5 milliliters per 100 milliliters, that there was a huge change in partial pressure that allowed that decrease in oxygen amounts, the release of that oxygen. So the blood would have to go from 100 mmHgs all the way down to 40 in partial pressure to release that 5 milliliters of blood. But what you notice is when it goes from 15 milliliters of O2 per 100 milliliters of blood to 10, that the partial pressure actually does not have to change as drastically to release that amount of blood. So when the blood is at a lower partial pressure, its partial pressure must fall only a little for it to release more oxygen. So you get a faster release of oxygen at lower partial pressures. And this faster release of oxygen at lower partial pressures is due to the binding of oxygen and the structure of hemoglobin in general. So we'll talk a little bit about how that occurs, but ultimately the release of oxygen increases as the partial pressure actually decreases. Now, when we look at different types of respiratory pigments in different types of animals, we can see a range of affinities for oxygen for the different respiratory pigments of these specific animals. So if we look at the different oxygen equilibrium curves for something like a seal versus human, you can see that their oxygen equilibrium curve, their amount of oxygen per 100 milliliters of blood based on partial pressure, their curves are very different. And what this has to do with is a couple different things, one of which is the respiratory pigments in these different animals have different affinities for oxygen, which allows them to upload and release at different rates. And this is due to different molecular versions of those respiratory pigments. So subtle mutational changes or differences in sequence can lead to differences in binding of oxygen. And also, depending on how many respiratory pigments are actually dissolved or found in the blood, that will influence how much oxygen carrying capacity that animal has. So in particular, when we look at the height of the curve, something like Waddell sills, the fact that they have such a higher oxygen carrying capacity, we're looking at 30 milliliters of oxygen per 100 milliliters of blood, you can see that it's much, much higher than what the maximum carrying capacity is of the human. And that has to do mostly due to higher levels of hemoglobin in the blood of the Waddell seal compared to the human. So different concentrations of the respiratory pigments as well as different affinities for oxygen can influence the oxygen carrying capacity and ultimately what partial pressure the saturation is reached for that particular, the oxygen saturation is reached for that particular animal. We can compare oxygen affinity from one organism to another, from one animal to another. We can look to see if their respiratory pigment has a higher affinity or not by looking at what's called a P50 value. And P50 value is basically 50% saturation. So 50% saturation of those heme groups, of the respiratory pigments, is means the partial pressure in which 50% of the respiratory pigments are saturated. That value allows us to compare the oxygen affinity of different respiratory pigments. And so if you look at this curve to the right, you'll notice that the p-value for the blue curve is lower than the p-value for the red curve. And what that indicates is the higher the p-value, the lower the affinity. And obviously, the lower the P50 value, the higher the affinity. So the particular respiratory pigments of curve 50, of the blue curve, has a higher affinity than the respiratory pigments that generate the red curve that's shown here. So ultimately, P50 values allow you to determine differences in oxygen affinity. Now, if the respiratory pigment has a lower affinity, then it fully loads at a higher partial pressure, but also unloads at a higher partial pressure. Higher affinity respiratory pigments fully load at lower partial pressures, but also have to unload at lower partial pressures. And you can see that based on these two curves here. If you look at it, you'll notice the lower affinity, which is here, actually has to reach to fully load. It fully loads at at higher partial pressures, which you can see here. It becomes higher partial pressure to actually reach fully loaded, or it actually unloads at a lower partial pressure. There are many factors that can influence the affinity of a pigment for oxygen, and one of which is cooperativity. We're also going to look at something called the Bohr effect, and then we're also going to look at effects of temperature on affinity and how it influences affinity for a pigment for oxygen. Now let's look at cooperativity first. In regards to cooperativity, when you have something like a hemoglobin molecule that's made up of 4 globin proteins, a tetramer, what happens in that type of molecule is oxygen affinity actually increases as each molecule, as each subunit of oxygen binds. So you get an increased affinity. You also get an increased unloading as those oxygens start to come off of a larger quaternary structure like that tetramer. When you see something like cooperativity and binding, meaning the more subunits that are binding to oxygen, the increase in affinity for the other subunits for oxygen, or in the reverse, it unloads faster, it results in what's called a sigmoidal shape of the binding curve. And this particular figure shows you that sigmoidal curve for human blood hemoglobin, which is a tetramer. And you can see that kind of S shape to the curve. That curve in that representation would tell you that there's cooperativity occurring with human blood hemoglobin. However, if we compare it to something different that doesn't have sigmoidal a sigmoidal curve, we can look at what's called a hyperbolic curve, which this would indicate that there actually is no cooperativity. And the perfect example of that is myoglobin, and that's because myoglobin is a single subunit. There are no other subunits that would ultimately potentially influence the structure of other subunits so that it could increase its binding affinity. So a monomer essentially would just is not going to have cooperativity, whereas the case of hemoglobin that's found in the blood, we see cooperativity due to those multiple subunits. And this has to do with binding of oxygen to one subunit causes conformational state changes in other subunits that actually allows for an increased binding of oxygen. So binding at one influences binding at the others, which allows for cooperativity. And we specifically see the sigmoidal curve when you have this cooperativity. And most hemoglobins have cooperativity, and you can see that on some of the previous curves. If you look at the curve looking at other animals and their oxygen equilibrium curves, you'll notice that most of them are sigmoidal in shape, which means that those respiratory pigments in those particular animals, such as the seal versus the human, etc., that there's cooperativity and binding of oxygen to those respiratory pigments. Now, what is the Bohr effect? The Bohr effect has to do with a shift in oxygen binding, and this is ultimately due to partial pressure of carbon dioxide as well as changes in pH. And this particular curve is showing you that effect in regards to pH. So when there is a decrease in pH, there is a decrease in affinity for oxygen. So something like hemoglobin, which is shown here, in decreased pH solutions, hemoglobin exhibits a decrease in binding of oxygen. And this has to do with some important changes to the structure of hemoglobin. When pH decreases, there's a higher hydrogen ion concentration, and the hydrogen ions actually get picked up by hemoglobin molecules. And when they do, it alters the structure in such a way that it actually can decrease the affinity for oxygen. And you can see that by looking at the change in pH from 7.6 to 7.2, and you can see that the curve actually is shifting right. That means the P50 values, if you take and determine those P50 values, which I'm doing at the moment, you'll notice that the P50 values increase as the pH decreases, which indicates a decrease in affinity. Now, what happens in regards to the partial pressure of CO2? As the partial pressure of CO2 increases, you see a decrease in oxygen affinity for respiratory pigments like hemoglobin. And so the same type of shift is observed that we saw with pH, except this has to do with CO2 partial pressure. So as the partial pressure of CO2 is increasing, if we look at our P50 values again, they are going up with increasing CO2 partial pressures, ultimately leading to or indicating a decrease in oxygen affinity. So why is the Bohr effect important? The Bohr effect is important because it helps enhance oxygen delivery in an animal. When we think about the tissues in general, the tissues are going to have a higher partial pressure of CO2, and the pH is going to be lower due to that increase in CO2. And this is going to allow for a decrease in affinity for oxygen for hemoglobin, which would be important because it's going to help lead to release of oxygen into the tissues. In the lungs, however, the partial pressure is lower, which subsequently leads to a higher pH, and that means that there's going to be a higher affinity for oxygen for hemoglobin in the lung, which is going to help promote oxygen uptake into the blood. So ultimately, we build up higher levels of CO2 in the tissue. That's going to help lead to release of oxygen, and we want to move or uptake oxygen in the lungs to the blood, and that lower partial pressure of CO2 as it leaves the blood ultimately is going to help promote uptake so that we can take oxygen to the body. So the Bohr effect obviously is going to enhance uptake and delivery of oxygen to all parts of the body. Temperature also influences oxygen affinity, and you can see that a increase in temperature, when we compare two different temperature amounts, when you increase temperature, it leads to a decrease in affinity of oxygen for the respiratory pigment. Now, besides carrying oxygen, respiratory pigments are also important for CO2 transport, as well as not just the respiratory pigments, but the blood is going to be important in CO2 transport, so it ultimately gets rid of CO2 waste. And when we think about carbon dioxide and how it's carried in the blood, there are three forms in which it's carried. The main form is it's actually carried as bicarbonate. That's about 70% of the CO2 is transported as bicarbonate, which is shown here. And this is actually catalyzed by an enzyme called carbonic anhydrase, and it's found in different locations to help generate bicarbonate from CO2 and water. A smaller portion of CO2 is dissolved in the fluid, and there's also some amount of carbon dioxide that's carried directly by the pigments themselves as a carbon amino hemoglobin percentage, which is 23%. So if we next figure, what you'll see is we have what's called the Haldane effect. And the Haldane effect has to do with there's a shift in carbon dioxide equilibrium in the equilibrium curve due to the oxygenated or deoxygenated state of hemoglobin. And from what you can see, when the blood is deoxygenated, there's actually a greater carrying capacity for CO2. When the blood is fully oxygenated, there's actually a decrease in the CO2 carrying capacity of the blood. So carbon dioxide concentration at a given partial pressure is higher in deoxygenated blood than in oxygenated blood. So ultimately, when deoxygenated blood leads to tissue, increase in CO2 levels are going to actually be able to move into the blood and help remove it from the body. So the Haldane effect is really going to help remove CO2 from the body. Now, when we look at the different forms of CO2 in the blood, these are the three different types that we talked about. So one type is shown here, which is hemoglobin can actually carry CO2 by hemoglobin actually being having certain amino acids actually bind CO2 and can help carry CO2 in the blood. The other main form of CO2 that's in the blood is, remember, bicarbonate, and that's about 70% of the CO2 is actually found as its bicarbonate version, which you can see there, and in different ways in which that bicarbonate's actually formed. And there's also small levels of CO2 actually dissolved in the blood as when it's carried in the blood. So there's three different forms of CO2 in the blood, which will help ultimately remove the CO2 from the body. Bicarbonate bound to the hemoglobin in the red blood cells, and then also a small level of CO2 that's actually dissolved in the blood. And this is the end of this lecture. So next Tuesday when we meet again, we are going to move forward and talk about circulation. This is the last in part that will be on the exam on Thursday. So everything covered through today's lecture that you're listening to right now, that is on the next exam. However, the circulation lecture that we'll talk about on Tuesday, this next coming Tuesday, will be on the last exam for the semester.